I'm presenting an experimental design question to test the role of a protein that our lab has identified called protein X. And we believe that protein X is required for non-homologous end joining. So protein X has some sequence conservation with other chromatin remodeling factors, and it also has a weak ATPase activity in vitro. So we hypothesize that protein X promotes NHEJ in the context of chromatin by removing nucleosomes from the ends of DNA double-stranded breaks. So just to give you some background on NHEJ, it is the predominant repair pathway of double-stranded DNA breaks in mammalian cells. So when there is a double-stranded break in your DNA, this is first recognized by the Ku heterodimer, which will bind to the free ends. And this will recruit other components that are required for NHEJ function that will combine to form the synaptic complex. And ultimately, this will result in ligation of the two free DNA ends by the ligase LAG4. And this will result in the repair of that DNA double-stranded break. So it's important to understand how NHEJ normally functions because it is a common target of chemotherapeutics. And as we all know, chromatin is the natural state of your genome. So it's important to understand how this process occurs in the presence of nucleosomes. So the assay that we'll be using to test the role of protein X is called the ensemble end joining assay. So this involves a linear piece of DNA that has sequences at the end called nucleosome positioning sequences. And this will allow us to assemble nucleosomes at the ends of this DNA. So we will assemble the nucleosomes onto this using a salt gradient dialysis. And this process will result in the binding of the nucleosomes to these nucleosome positioning sequences. So we can now add this into an in vitro assay with the NHEJ components to observe how the presence of nucleosomes interferes, if it interferes with NHEJ. So the assay will take our linear piece of DNA, and to confirm that we can actually reconstitute NHEJ in vitro, we will combine purified NHEJ components. An alternative would be to use a cell-free extract that should be competent for NHEJ. But the problem with that alternative is that cell-free extract may contain other factors that could complicate our analysis, including proteins that have a similar function to protein X. So the cleaner alternative is just to use purified components. So in the presence of the purified NHEJ components, we predict that our linear piece of DNA will be circularized in the absence of nucleosomes. And we can read this out on a DNA gel that we will visualize with an intercalating agent such as cybergold or ethidium bromide. So if we run our linear strand of DNA that has not been mixed with the NHEJ components, we should see a fast migrating band on our gel corresponding to the linear species. In contrast, if our reaction is competent for NHEJ, we should see the circularization of the product of the plasmid, resulting over time in the accumulation of a larger piece of DNA that migrates more slowly in the gel, corresponding to the circularized plasmid. It's also possible that since these NHEJ components will ligate together any free DNA pieces that they find, that we could also see the appearance of higher order species that would correspond to multimers of our linear piece of DNA. So So this is over time. And this functions as a positive control to confirm that we are able to reconstitute non-homologous end joining in vitro with our linear substrate and purified NHEJ components. In this case, a negative control doesn't necessarily make sense because it's not surprising that if we don't add all of the NHEJ components to the mixture, that we would not see NHEJ. So in that case, a negative control for this reconstitution experiment doesn't necessarily make sense. So now that we've confirmed that we are able to perform NHEJ in vitro, we will now test the role of protein X. So we have a couple of reaction conditions. The first one will be our linear piece of DNA with our nucleosome positioning sequences, but in the absence of nucleosomes. So again, if we add the NHEJ components, we expect to see the circularization of our plasmid. And I'll draw our results out here. So in the presence of NHEJ components, but in the absence of nucleosomes, we expect to see the formation of a circularized plasmid. In contrast, if we assemble nucleosomes onto our linear substrate first, we predict that these will interfere with the non-homologous end joining process and that we should see the formation, or we should still see our linear product at the end of the reaction. And this would run, again, as the faster migrating species. However, if we do see the formation of a circular product, that would indicate that potentially our nucleosome assembly reaction did not work. And in order to test that, we could perform an MNase digestion reaction to confirm whether or not the nucleosomes were actually assembled properly onto the DNA. And that would explain why we continued to see circularization of the plasmid. So our next reaction is going to include protein X to confirm or to test our hypothesis that protein X is able to facilitate non-homologous end joining even in the presence of nucleosomes. So we will again assemble our nucleosomes onto the linear substrate using salt gradient dialysis. We will add in our NHEJ components, and we will also add in protein X. And if we do see circularization of the plasmid, that would be consistent with a role for protein X in facilitating non-homologous end joining by removing nucleosomes from the free ends of the DNA. Oh, this was plus NHEJ and plus nucleosomes. If we add in protein X now, in the presence of nucleosomes, we expect to see the formation of our circularized plasmid. The final reaction condition that we're going to test is to see if the ATPase activity of protein X is required for this role in promoting non-homologous end joining. So we will again assemble nucleosomes onto our linear substrate, add in the NHEJ components, but in this case, we're going to use an ATPase dead form of protein X. So this is a catalytic mutant that is unable to hydrolyze ATP. An alternative would be to remove ATP from the reaction conditions, but in this case, the actual ligation of the plasmid requires ATP, so that's not a possibility for us to test. If the ATPase function or activity of protein X is required for its role in promoting non-homologous end joining, we would expect to see, again, our linear piece of DNA, consistent with just adding in nucleosomes and just the NHEJ components. So on our gel, if we have an ATPase dead form of protein X, In the presence of the non-homologous end joining components and nucleosomes, we would again see a linear product. And this result would be consistent with the role for protein X using its ATPase activity to remodel the chromatin at the ends of DNA double-stranded breaks in order to facilitate non-homologous end joining.